Hello friends, today's my discussion is on neuromuscular junction. So after the completion of this discussion, the students must be able to draw the systematic diagram of neuromuscular junction and the structure of neuromuscular junctions and describe the events of the neuromuscular transmission at neuromuscular junction. So these all are learning objectives. So what is the definition of the neuromuscular junction? So neuromuscular junction, it is the junction between the terminal branch of the nerve fiber and muscle fiber. Structure of the neuromuscular junction can be broadly divided into three parts: presynaptic, synaptic and postsynaptic portion. Now presynaptic portion. So the neurons innervating the skeletal muscle fibers are known as motor neurons and that have their cell body in the anterior horn of spinal cord or in the brainstem. So this is the cross section of the spinal cord and this is the anterior horn. So this is the motor neuron and that have their cell body in the interior horn of the spinal cord or brainstem. So sensory impulses that arises from the receptors that are located in the skin or mucous membrane and the impulses passes through the sensory neurons and they relay in the posterior horn and this one is a interneuron or relay neuron and then the impulses passes through the motor neuron and then the effectors are muscle fibers or muscle. So the neurons innervating the skeletal muscles are known as motor neuron and that have their cell body in the anterior horn of the spinal cord or in the brainstem. And their axons are myelinated and they are largest diameter axon in the body. Then what are the features of the presynaptic portion of the neuromuscular junction? So as the neuron of the motor neuron that approaches to the muscle fibers, it loses its myelin sheath and they are divided into several branches and that have a diameter of about 2 micrometer and that are known as axon terminals. So these are the motor neurons. When they reaches the muscle fibers, they lose their myelin sheath and they are divided into many terminal branches and that is known as axon terminals. Now the terminals that are covered by Schwann cells that are known as teloglia. So this is teloglia. Axon terminals that are, these are branch of axon terminal and this is axon terminal that are covered by Schwann cells that is known as teloglia. Now each terminal that forms a junction with the single skeletal muscle fiber midway along its length. So each muscle fiber is supplied by one motor neuron terminal. The motor neuron including its axon and axon terminals and the muscle fibers supplied by it are known as motor unit. So these are the motor neuron including its axon and its all axon terminals and all the muscle fibers that are supplied by it, that all together is known as motor neuron. In my next lecture, I will discuss about the motor unit in detail. Then each terminal is expanded at its end to form a knob-like structure that is known as synaptic knob or terminal button. So this is a knob-like structure that is known as synaptic knob and it contains a plenty of mitochondria and synaptic vesicles. The terminal button that lies in the groove in the surface of the muscle fiber. So this is the muscle fibers and this terminal button or synaptic knob that lies in the groove in the surface of the muscle fiber but outside the muscle cell membrane. Now, the presence of the large number of the mitochondria indicates the higher metabolic activity at terminal region. The vesicles that are collected around a specific points that is known as active zone. So suppose these all are vesicles, they are collected at specific point and that is known as active zone. This is known as active zone. And the membrane at the active zone that is modified to form dense bar. So this is a membrane that is modified to form the dense bar like this, and that contains a numerous voltage gated calcium channel, and this channel mediate acetylcholine release. Now the membrane of the nerve ending that is known as presynaptic membrane. So membrane of this nerve ending that is known as presynaptic membrane, and the membrane of the muscle fiber that is known as postsynaptic membrane. So the space between the presynaptic and postsynaptic membrane that is known as synaptic cleft. So what is the synaptic cleft? The space between the pre and postsynaptic membrane that is known as synaptic cleft. And synaptic cleft, it is about 40 to 100 nanometer wide. And the muscle fiber is covered by a layer of amorphous connective tissue that is known as basement membrane or basal lamina, that consisting of collagen, glycoprotein and other extracellular matrix protein. The basement membrane in the cleft contain the enzyme acetylcholine esterase. So this is the synaptic cleft and it contains a acetylcholine esterase enzyme. Acetylcholine esterase rapidly hydrolyzes acetylcholine into acetate and choline. The basal lamina helps organize the presynaptic button with the postsynaptic junctional folds. So these muscle membranes are folds into a many folds that is known as junctional folds. Now postsynaptic portion. So postsynaptic portion of the neuromuscular junction is known as end plate membrane or motor end plate. So this all is known as motor end plate. This is the part of the sarcolemma that lies directly under the terminal button. So this is a terminal button or synaptic knob. So the part of the sarcolemma means membrane of the muscle fiber that lies directly under the terminal button that is known as motor end plate. The area of the end plate membrane increases many times as it is thrown into several folds known as junctional folds. So this all is junctional folds. The end plate membrane contains a acetylcholine receptor which are concentrated at the crest of the junctional folds. So in over the junctional folds there are a acetylcholine receptors are present, and it also contains a voltage gated sodium channel. Acetylcholine receptors. So now about the acetylcholine receptors that are present at the neuromuscular junction, and they are nicotinic type. An average end plate contains about 15 to 40 million acetylcholine receptor, and which accounts for about 10,000 acetylcholine receptor per square micrometer of end plate membrane. Now acetylcholine receptor, it is a protein and it is a chemically gated ion channel. And whenever the two molecules of the acetylcholine bind to it, it undergoes a configurational change. And this channel is blocked by alpha bungarotoxin. So acetylcholine receptor, that is a protein and it is a chemically gated channel and that undergoes. These all are acetylcholine receptors. And when the two molecules of the acetylcholine bind to these receptors, there is a configurational changes occurs in that channel. And this channel that is blocked by alpha bungarotoxin. Now, because of its large pore diameter, it is permeable to both ions, sodium and potassium. But as the electrochemical gradient is favorable for the sodium entry, more sodium moves in than potassium coming out. So, result in net sodium influx. So, in this figure, in A, that is a closed state, and B, that is a open state. And conformational change that occurred due to binding of the two molecule of the acetylcholine to the binding site. Now acetylcholine receptor that allows the passage of only cations because the anions are repelled by the fixed negative charge is present in it. Now there are a certain chemical transmissions that occurs at the neuromuscular junction. So the events are, first of all, there is a release of acetylcholine, action of acetylcholine, then development of end plate potential, development of miniature end plate potential, and then destruction of acetylcholine. So first of all, release of acetylcholine. So when the action potential reaches the axon terminal, so this action potential when reaches the axon terminal, it opens the voltage gated calcium channel in the membrane of axon terminal and calcium from the extracellular fluid enter the axon terminal. So this causes activation and opening of the voltage gated calcium channel and that leads to calcium influx. Increased cytoplasmic calcium concentration increases the movement of the microtubules and microfilaments that leads to migration of neurotransmitter vesicles to the inner surface of the presynaptic membrane. So the vesicles that fuse with the membrane and discharge their contents into the synaptic cleft by exocytosis. Now the various membrane proteins are present in the vesicular membrane as well as in the neuronal membrane that are involved in the process of fusion. So action potential reaches the axon terminal, then the voltage gated calcium channel open. Now the synaptobrevin or synaptotegmin that are a protein and that is present in the vesicular membrane means on the membranes of the vesicles. This protein synaptobrevin and synaptotegmin are present and these all are proteins are calcium sensitive. Now there is another membrane, axonal membrane. So here syntaxin and SNAP25 these proteins are present. Now when the action potential that reaches at the presynaptic axon terminal, depolarization occurs that opens the calcium channel in the plasma membrane. So here plasma membrane the calcium channels are open. So calcium that enters into the cytoplasm. Now this calcium that binds to a calcium sensitive protein that is known as synaptotegmin and that is present on the membrane of the vesicles. Now many synaptic vesicles are already docked at the correct area of the presynaptic membrane before the arrival of the action potential. So many synaptic vesicles that are docked at the active zone of the axonal membrane. Now at these docking sites, the vesicles are attached by protein. Which protein? Syntaxin and SNAP24 that are present on the axonal membrane. These proteins are present on this membrane, axonal membrane. Now when the calcium that enters inside, that binds with the synaptotagmin. Now this complex that are docked into this site. This is a docking site. So that form a fusion complex that is associated with the presynaptic membrane. So docking that involves a snare complex of protein that bridge the vesicle membrane and plasma membrane. So this, at first there is a docking occurs and then there is a fusion of vesicle membrane and plasma axonal membrane occurs. So snare protein that include one in the vesicle membrane, that is a synaptobrevin 2, and two anchored in the plasma membrane, that are syntaxin and SNAP25. Now through a mechanism not fully understood, there is an interaction between the snare protein complex and, means this is a snare protein complex, this is a snare complex, and calcium synaptotagmin complex leads in less than a millisecond to fusion of the vesicle and plasma membrane. So there is a formation of the pore occurs. There is a fusion of vesicle membrane and axonal membrane. So there is a pore formation occurs and there is a exocytosis of neurotransmitter acetylcholine. So here you can very well see this is a membrane of the vesicles and this is a axonal membrane and there is a snare proteins are present and there is a synaptotagmin and calcium complex. So there is a fusion of this vesicle membrane and axonal membrane, and finally there is a pore formation occurs and there is a exocytosis of the acetylcholine occurs. The scientists who discovered how the synaptic vesicles dock and release their neurotransmitters were awarded the 2013 Nobel Prize in Physiology or Medicine. By exocytosis, the acetylcholine diffuses through the presynaptic membrane and enter the synaptic cleft. Each vesicle contains about 10,000 acetylcholine molecules, and at the same time about 300 vesicles open and release acetylcholine. So first events that occur at the neuromuscular junction is release of acetylcholine. Then second is action of acetylcholine. So after entering the synaptic cleft, acetylcholine molecules that bind with the nicotinic receptors present in the postsynaptic membrane and form the acetylcholine receptor complex. That increases the permeability of the postsynaptic membrane for the sodium ion by opening the ligand-gated sodium channel. Now sodium ions from the extracellular fluid enter the neuromuscular junction through this channel. And there sodium ions alter the resting membrane potential and develops the electrical potential that is known as end plate potential. Then what is the end plate potential and what are the properties of end plate potential? So end plate potential, that is a graded potential and it is not the action potential. So what is the significance? So end plate potential, that is localized and non-propagative and it does not obey all or none law, but it causes the development of the action potential in the muscle fiber. Then development of miniature end plate potential. So miniature end plate potential, that is a very weak end plate potential in the neuromuscular junction, and that is developed by release of only a small quantity of the acetylcholine from the axon terminal. And each quantum of this neurotransmitter that produces a weak miniature end plate potential, and the amplitude of this potential is only up to 0.5 millivolt. Miniature end plate potential cannot produce action potential in the muscle fiber, but when more and more quanta of the acetylcholine are released continuously, the miniature end plate potential are added together, and finally that produce end plate potential, and that result in action potential in the muscle fiber. Then destruction of acetylcholine. So acetylcholine released into the synaptic cleft, that is destroyed very quickly within 1 millisecond by the enzyme acetylcholine esterase. However, the acetylcholine is so potent that even this short duration of 1 millisecond, that is sufficient to excite the muscle fiber. Rapid destruction of the acetylcholine has got some important functional significance because it prevents the repeated excitation of the muscle fiber and allows the muscle fibers to relax. Then reuptake process. Reuptake is a process in the neuromuscular junction by which a degraded product of neurotransmitter acetylcholine that re-enters the presynaptic axon terminal where it is reused. Acetylcholine esterase splits or degrades acetylcholine into inactive choline and acetate. Choline is taken back into the axon terminal from the synaptic cleft by the reuptake process. There it is reused in the synaptic vesicles to form new acetylcholine molecule. So these are the sequence of the events during the neuromuscular junction transmission. So whenever the axon potential arrives, there is a opening of the voltage gated calcium channel. So there is an entry of the calcium ions. So there is a opening of the vesicles and there is a release of acetylcholine. Now this acetylcholine that enters the synaptic cleft, and where it at the postsynaptic membranes there are so many acetylcholine receptors are present, and this acetylcholine that binds with the receptor, and there is a formation of acetylcholine receptor complex. Now this complex are responsible for the opening of the sodium channel. So there is an entry of the sodium ions and there is a development of the end plate potential, and this end plate potential that is responsible for the generation of the axon potential in the muscle fiber, and then there is a muscle fibers respond by contraction, and there is a excitation contraction coupling process occurs. And this is the video for the events that occurs at the neuromuscular junction.
A skeletal muscle contracts only when stimulated by nerve impulses from a motor neuron. Without innervation, muscles become paralyzed and eventually atrophied. The axon of a motor neuron usually gives out many branches, supplying multiple muscle fibers. These fibers contract in unison when activated and constitute a motor unit. A motor unit can contain anywhere from just a few muscle fibers to thousands of them. Small motor units are found in muscles that require finer control, for example, muscles that are responsible for subtle movements of the eyes. Large motor units are found in larger muscles that require strength, such as muscles of the arms and legs. Muscle fibers of the same motor unit are usually not clustered together, but scattered throughout the muscle. This way, stimulation of a motor unit has a slight but uniform effect on the whole muscle, and not causing a small, localized twitch. The strength of a muscle contraction is determined by the number of motor units that are activated at one time. Even at rest, most muscles are in a partial contraction state called muscle tonus, which is maintained by alternating activation of a small number of motor units. The connection between a motor neuron and a muscle fiber is called a neuromuscular junction, which is basically a chemical synapse between the nerve terminal and a specialized area of muscle cell membrane called the motor end plate. When an action potential reaches the nerve terminal, it causes the release of the neurotransmitter acetylcholine into the synaptic space. Acetylcholine then binds to nicotinic receptors on the end plate. Nicotinic receptors are ligand-gated ion channels. Upon binding to acetylcholine, they open to allow sodium to enter the cells, depolarizing the cell membrane, producing the so-called end plate potential. An action potential is generated in the muscle cell only when the end plate potential reaches the threshold required to activate voltage-gated sodium channels located outside the end plate in the neighboring membrane. When activated, these channels allow faster influx of sodium, further depolarizing and eventually reversing the polarity of the cell membrane. At this point, voltage-gated potassium channels open for potassium to move out, quickly returning membrane voltage to its original resting value. Once generated, the action potential spreads like a wave, thanks to similar voltage-gated ion channels located throughout the muscle fiber. The action potential also runs deep into the fiber via T tubules to reach the sarcoplasmic reticulum. Here it activates voltage-gated calcium channels, releasing calcium from the sarcoplasmic reticulum into the cytosol of muscle cells. Calcium then sets off muscle contraction by the sliding filament mechanism. This mechanism is described in another video. Another important component of the neuromuscular junction is the enzyme acetylcholinesterase. This enzyme removes all acetylcholine molecules that do not immediately bind with the receptor and those that are done activating a receptor. The enzyme action essentially terminates synaptic activation, giving the muscle time to relax and thus preventing continuous contraction that would result in muscle spasms. Substances that cause muscle weakness or paralysis do so by interfering with the function of neuromuscular junction. Botulinum toxin prevents acetylcholine release from the presynaptic side of the junction. Some other toxins attach to nicotinic receptors, blocking acetylcholine from binding but do not open the ion channel. Certain drugs lodge into the channel of nicotinic receptor, blocking the passage of sodium. All these substances prevent activation of muscle cells and cause flaccid paralysis. On the other hand, some pesticides inhibit acetylcholinesterase, preventing degradation of acetylcholine, causing continuous activation of muscles. That's how they induce muscle spasms and cause spastic paralysis.
So this is the another figure for the transmission occurs at the neuromuscular junctions. These all are events occurs in a sequence from 1 to 8. So you can read from this. Thank you very much.